Okay, so they give me a thumbs up, so I guess that means I get to start. So first off, some of you came in a little later. Up here by John Ione on the seat right here, we have these little slide rules. We're going to be using these today. So if you want to come up and grab one, you can. If you want to grab one afterwards, basically it's the ISO 10993 chart in a slide rule form. So there's some other ones in the back that have to do with like packaging and stuff. Those are recommendations that Nelson Labs gives, but this right here is kind of the Biol chart that we all use for Biocomp. So if you want to grab these, we'll be using them a little bit today and during this workshop. And in fact, I kind of want to make this a workshop. This is something that I really enjoy talking about. The reason, for me, it can seem a little simple because we're going to be talking about the basics of biocompatibility. And a lot of people do biocompatibility. But what I've learned talking to people is that they get thrown into biocompatibility and they really don't know what they've gotten themselves into. And all they start doing is just trying to figure out where they're going. And they end up maybe making some mistakes along the way. So I'm hoping that what we can do is kind of break it back down to its simplicity, the concept of why we run biocompatibility, and kind of help you make decisions on the testing that best represents the end use of your device. So the first thing I wanted to kind of touch on today is, what is biocompatibility? I get this question all the time, like, what are we doing? We're just doing animal tests? Is that biocompatibility? Or what does it entail? Well, here is the IUPAC definition, which is the ability of a material to perform with an appropriate host response in a specific application. And for me, that makes sense, but I gave that to someone at Nelson Labs that does micro testing, and they went, what? That didn't make any sense. Really, what we're trying to see is, will your device cause any harm? Okay? The whole reason we got into this, at least I believe so, the reason I got into this industry was to help people. And that's really what I'm passionate about. I really want to help get or devices to the market that will help people overcome sicknesses or injuries and make their lives better. Well, that's great, but if the device we're trying to make that does to do that ends up hurting someone, then we're contradicting that aim. So biocompatibility, really what it does is it helps screen your materials to see if there's any kind of impact in the end user. It's not a catch-all, right? We're trying to do our best we can to screen for your device to make sure the impact is minimal. And basically the whole concept is that the benefit of the device outweighs any risk associated with that device in the body. So there are some tests that we use. There's a lot of tests we can use for biocompatibility. We're going to cover a couple of them really high level today, some that we use, but more importantly, we're going to go over what tests we need to do for what devices. How do we categorize our devices today? So the first three tests, there are tests that we call the big three, and that's the cytotox, the sensitization, and the irritation. And we call them the big three because those are three tests that almost every medical device manufacturer has to look at performing according to the ISO 10993 chart. And then we have other tests that we're not going to spend any time on today, but we have other talks that will go over them. But it has to do with systemic toxicity, genotoxicity, implantation. There's a whole range of testing. But the whole concept is we try to screen different endpoints to look for safety of a medical device. So the first thing we're going to cover today is what I like to affectionately call your best friend and your worst nightmare, and that's the cytotoxicity test. And the reason why I call cytotoxicity your best friend and your worst nightmare is because historically, this is the most sensitive test we have for biocompatibility. In fact, if you look through the last 12 years at Nelson Labs, if you fail a test, 93% of the time, that failure occurs in cytotoxicity, okay? So that doesn't mean that this test will fail 93% of the time. That would be outrageous. But what it means, if you're going to fail, this is probably your worst case, right? This is probably what's going to happen. Now, that's why it's your enemy. It's because if you do it long enough, you're probably going to see a failure. But it's your best friend because it's the quickest and the least expensive biocompatibility test we have. Okay? So it makes a great screening test. In fact, that's really its intended use, is to kind of screen for potential impact later on. So we can use that for changes of the device to help screen the potential impact of a change of a process like H2O to hydrogen peroxide gas to EO gas. We just want to kind of screen for potential impact of a proposed change. Cytotoxicity is great. It costs about $150 to $170 depending on the lab you go to, and takes about 5-7 days to 2 weeks depending on the lab you go to. So it's a quick, relatively quick and inexpensive test, and it just happens to be the most sensitive, okay? So for this test, this is my dashboard, I call it. It kind of gives you a snapshot of information about each of these tests. So up in the top right, we have sample size requirements, how much sample required for these tests, the turnaround time, and that's specific to Nelson Labs. Each lab has a little bit of variation, but that's Nelson Labs turnaround time. And then some usual problems, okay? For the cytotoxicity, latex is what we use for a positive control, okay? So latex kind of does pretty bad things to cells. So if you have latex in your device, it's gonna fail. But that doesn't mean you can't use latex, right? I mean, how many decades have doctors been using latex gloves? What it means is we have to look at it from a risk assessment of why you failed that test or why you might fail that test. We might want to remove the latex to see if there's any other components in your device that are cytotoxic, and then justify why accepting that toxicity of that latex. That also goes for other natural rubbers. Silver, copper, zinc, all those metals have +2 ion interactions with the cells and cause a hypotonic solution, which is pretty bad for cells. It just happens to be a lot of inks have metal components in them, like silver, copper, and zinc, right? So if you have an ink that is leaching off a device, you could have problems there too. Another usual problems we see with cytotoxicity is leaching of adhesives. And we affectionately call it at Nelson Labs the smell test. We open up a device bag and all of a sudden you get that really strong chemical smell. Well, if you're getting that strong chemical smell, guess what's coming off in the extraction media? Strong chemicals, right? And just because an adhesive says it's medical grade, you have to look at why they're claiming medical grade and how they tested that medical grade. Sometimes when we test medical grade or adhesives for medical grade, how they perform cytotoxicity is they do a very thin layer on a substrate like a stainless steel or even a polypropylene or a glass slide, but they use a very thin layer and they cure it all the way. But you guys don't always use medical adhesives that way. Sometimes you put a big goop of it on both ends and put it together, and sometimes it doesn't cure enough. And so you might have some failure based off how much of the chemicals are coming off the device from not being cured all the way. So these are some of the issues we see in cytotoxicity. Recently we've seen some issues with alternative plasticizers. So if you guys know what a plasticizer is, it makes a hard plastic soft, right? For the longest time, DHB and BPA was used. Now those are almost swear words in the industry, depending on where you're submitting your regulatory arena. And so they've started going away to some citrate-based plasticizers or some alternative plasticizers, and sometimes we're seeing issues with those in cytotoxicity. So some things to think about when you're running that test. The next test we're going to briefly cover is sensitization. Now, sensitization is the longest and the most expensive of the big three tests. It looks for something called delayed hypersensitivity. And the best way to explain it is poison ivy. Okay, so the first time you contact poison ivy, nothing happens. You don't even realize you contacted the plant. But your body does. It remembers those toxins. And so the next time you touch the plant or the third time you touch it, you break out. And so for that reason, this test takes forever. It takes about 8 weeks to run because we have to repeat expose the animal to an extract to look for that sensitization reaction. So we just can't expose it once and say, oh, look, nothing happened, we're fine. We have to do that repeated exposure. And for that reason, this is a long and expensive test. The other kind of downfall to the maximization is it's qualitative data only. We're looking for redness and swelling, what everyone knows is an allergic reaction. Okay, so it's a very quantitative test, but it does take quite a bit of time and a lot of sample. If you see, it takes 6 times more sample than the cytotoxicity test because we're extracting in a polar and non-polar media, and there's 3 exposures. So there's 6 times more sample required for the sensitization. For this reason, for the cost and the turnaround time and the sample amounts, sometimes this test can be a bad word in some medical device manufacturers. Oops. Went the wrong way, excuse me. The next one we're going to talk about is irritation. And irritation is a one exposure immediate response, an immediate immune response to some kind of antigen, okay? And the irritation, because it's an immediate one exposure response, It's very similar to the outlook of a sensitization test, but instead of that repeated exposure, we're looking for the first time immediate response. And so for this reason, it takes about 4 weeks instead of the 8 to 9 weeks. But the other thing you got to remember about irritation is that the specific part of the body can have a different irritation response, okay? So if you put something in your eye and you put something on your skin, the irritation response could be totally different. It could be a non-skin irritant, but it could be an ocular irritant. For this reason, we have specific tests designed for specific parts of the body. We have an ocular irritation, we have mucosal irritation, vaginal irritation. All these can be specific to the intended use of your device. The general kind of tissue and blood contact test that we run is called an intracutaneous reactivity. And that's the irritation test that's most commonly run. And that's one that's injected intracutaneous. We tend to kind of be very duh with our names of our tests. So intracutaneous reactivity means we're injecting the extract intracutaneously. But it's very quick, well, relatively speaking to the sensitization, about 4 weeks, and it's because we're looking for that immediate response. The next test we're going to talk on very high level is acute systemic toxicity, okay? This is up to this point, you got to choose what tests you want to run based off the, you can either do like the vaginal or mucosal irritation or the intracutaneous irritation. You just choose which one fits best. In this category, there's 2 tests, acute systemic injection and material-mediated pyrogen. Both these tests are usually required or required to justify why you did not perform, okay? Both of these are really quite simple though. They're quick injections in either in a mouse or a rabbit, and you're looking for an immune response. So for a systemic injection, you're looking for a biological response. Do they become lethargic? Do they start to, do they gain weight appropriately? And for material-mediated pyrogen, you're looking for a fever, which is a pyrogen. So you're looking for a response in that animal. Okay, so now we kind of very high level and very quickly covered those tests. The reason I wanted to do that is because in the first part of test selection, I kind of wanted to hit on USP Class 6. And the reason I do this is because it starts to become a bane of my existence when we start looking at biocompatibility. And that's because I get in a device and we run it through the testing and it fails. So I call up the sponsor. I'm usually the one that deals with the failures, and so I call up the sponsor and I say, your device failed. And they go, that's impossible. It's made out of plastic that's USP Class 6. Okay, that's great, but do they really know what USP Class 6 is? Okay, so just so you kind of know what USP Class 6 was designed for, it was designed for raw material plastic, specifically for container closures. Okay, and this is done in the USP Section 88. It's where you find USP Class 6, and it's really different than the ISO 10993. There's similarities, but there's a big difference. These are the 3 tests that are included in a USP Class 6 test regimen. So you have the irritation test, the systemic injection, and then a 1-week implant, okay, with no histopathology. And that's your USP Class 6. So you do this, you pass it, you get a little certificate that says USP Class 6 certified, and they hand that out like candy, right, on Halloween. Who can tell me what's missing though? If I had a gold star, you would get it. That's right. Cytotoxicity, right? Cytotox is the most sensitive test we have. And we're not even including it in USP Class 6. So lots of times they'll fail cytotox and they say, that's impossible. It's USP Class 6. And I have to break it to them. It doesn't include cytotox in a USP Class 6. Every time Nelson Labs quotes a USP Class 6, we always quote a cytotoxicity too, because for us, it makes no sense to screen a material if you don't have cytotoxicity, okay? But be careful, when you're looking at raw materials, just don't look at that certification and go, we're good. Or just don't look at its medical grade, because what does that mean, right? What does medical grade really signify? So instead of just accepting whatever they claim it to be, go in and look at what testing they performed, look how they performed it, be educated on what testing is needed for your device, and make sure your raw materials are meeting those classifications, okay? So that means you might have to require them to do a cytotox if you're concerned about it. Some people just take it and cross their fingers, put it in the validation testing and hope, okay? But I wanted to let everyone know that USP Class 6, although it's a good screening test, it's not the end all, because there are tests, they don't include a sensitization either. So there are some tests that a medical device has to perform that USP Class 6 does not cover. So this is actually the USP Class 6 chart. There's actually 6 classes. You can be USP Class 1 if you really want to. And so I throw it in there depending on how many tests you run. You get a certain classification. So USP Class 6 has those 3 tests: the systemic injection, the irritation, the implantation, and then the extraction fluids for each one is listed there. So, and if you want a copy of this presentation, I'm more than happy. You can just give me your business card. I'll send it out to you. I'm not so careful with these. I probably should be, but I'm not. Okay, so the last test we're going to hit on is the implantation. Okay, so the implant is a very fun test. I guess the best for me is because it gives me a lot of information when you're looking at a biocompatibility response. And the reason why is we're putting this sample into the muscle, most of the time into the muscle of a rabbit, and we're leaving there for a certain amount of time. But then we take a histopathology of that implant site, and we really get to see what kind of interaction the body has with that material. Is there something happening with the cells around it? Is the body fighting it more than the negative control? What's really happening with that sample? And it gives us a really good information. So the implantation is actually, in my opinion, a very useful test to help determine true biological impact. We can do the test either intramuscular or subcutaneous, which means we can either put the device in the muscle or we can put it underneath the skin of a rabbit. My recommendation is always intramuscular. The FDA released in their newest draft that they prefer intramuscular because it's more conservative. If you have to do subcutaneous, we can talk about it, but intramuscular is by far the most common. Okay, so now we get to the part, and we covered that, probably the reason why you guys are here, because the name of the presentation is determining a test strategy, and that's why we're going to use these little cards. Once again, if you didn't get them, they're up here, but this is the testing category. And we're going to go over the chart really fast. So this is the chart that most people are familiar with. We just broke it down into a slide rule. We're going to go through the different categories really fast, okay? The first one is on body contact. So we have 3 major categories. We have surface device, external communicating, and implant. The only one that's somewhat confusing here is an external communicating device. I think everybody knows what a surface device and an implant is, right? But external communicating is a device that's outside the body and inside the body at the same time. Best example is like a delivery device or a surgical tool, where the surgeon's holding onto a handle on the proximal end, the distal end's inside the body. So it's outside the body and inside the body at the same time. External communicating. Each of the 3 categories have subcategories. So for surface, we have skin, mucosal membrane, and breast are compromised. Pretty self-explanatory. The next one, once again, is the one that I see a lot of people get confused at, which is blood path indirect, tissue bone dentin, and circulating blood. Now, a lot of people, when they get into the tissue, they think, oh, there's blood present. That means my device is blood path indirect, okay? That's not what it means. We know that when your device is in the tissue, that there's blood there. We know that there's interaction with the blood, but the question you have to ask yourself is, does that blood get recirculated? Does it go throughout the body or is it waste? Because when we start looking at the difference of testing, for blood path indirect, we have hemocompatibility testing. That's thrombosis, that's hemolysis, that's complement activation. Well, if that blood's not reused, then if it hemolyzes, we don't really care, right? So if that blood gets recirculated, then it's blood path indirect. The best example is an IV bag, okay? So an IV bag is sitting outside the body, and chemicals from the bag is leaching into the fluid. And that fluid goes down and goes into the vascular system. That bag is not sitting in the blood path directly, but it's indirectly communicating with the circulating blood, okay? So that's the best example for, in my opinion, for blood path indirect, where a delivery device for a stent, the distal end's in the circulating blood path, so therefore it's direct blood path. Everything in the tissue, if that blood's just kind of waste blood, then we would do tissue or bone dentin contact. Tissue bone, for the implant, we have tissue, bone, and implant. So pretty self-explanatory. The next one I always get questions too, which is the duration of contact. So according to ISO, if it's less than 24-hour contact, it's an A. If it's between 24 hours and 30 days, it's a B. Anything over 30 days is permanent. And yes, we think more than 30 days is a permanent contacting device. What people get confused at though is just not one contact. It's repeated contact you have to think about too. Does this device get reused over and over and over again? If it does, you have to add up the total contact time, and that's your contact duration. So some people say, oh, it's only used for an hour, but it's used for an hour every day. And therefore, if it's over that space of time, it could be prolonged or permanent, depending on how long it could be used, okay? So this is actually the FDA version of the chart. It's off the G95, which it could be going away soon. They have a new draft out, but the chart's the same. So, okay, so let's have some fun. Fun for me, at least. I don't know about you guys. Let's see how we can categorize some of these devices, okay? So go ahead and use your nifty new charts, and we're gonna go through each one of these together and see how we categorize it. Each one of these are real life examples, okay? So the first one's a stent and delivery device. What do you guys think? First off, I always ask, do you test the stent and delivery device together or separate? Separate, that's right. The reason why is because we test these off a surface area, okay? So if we combine the stent and its delivery device together, how big is the stent? Right? The delivery device? So when we include the stent, we're really not testing the stent at all. If you're going to test them together, you're probably going to have to extract it in the same amount of fluid as one stent. And that's going to be very hard to extract a whole delivery device in the amount of fluid for one stent, right? So we want to always test them separate. Okay, so let's go the stent first. What do you guys think? Implant, right? Circulating blood, permanent, pretty easy. What about the delivery device? External communicating, circulating blood, hopefully less than 24 hours if your surgeon knows what he's doing, right? Okay, so that's pretty easy. I gave you a softball for the first one. The gauze packing strip. This is a packing strip that you have an open wound, and they don't want to close up the wound yet until everything looks good, so they pack it full of this gauze. And then once the gauze is saturated, they remove it, put some new gauze in until the surgeon's ready to close up the wound. Okay? Whoops, wrong way. What do you guys think? This one's a little harder. Okay, I hear a lot of times surface device, breached or compromised, right? That's how I categorized this device about 6 years ago. The FDA categorized an external communicating tissue bone denting, okay? Now, if you look at it, if you look at the big three, or if you look at tissue bone denting limited, you're looking at the big three. If you look at breach or compromised surface, you're looking at the big three. So the testing doesn't change. There's another thing that happened though. The company categorized it as 24-hour contact. The FDA said, well, how long can it be taken out and put back in? And the company said, well, as long as the surgeon thinks it needs to be done. So the FDA says, well, fine, that's permanent contact because it repeated a use. So they had to go back and put a limit on how long it could be used on their label. And then you always have the question about off-label use and things too. So that gets complicated. We're not going to get into that today. But that's some way they try to justify it. But you have to think about the repeated use. And in this case, it ended up being the FDA wanted permanent. They had to go along and get a prolonged contact device. Okay, this one, condom. What do you guys think? Surface device, mucosal membrane, less than 24 hours. 99% of the time, that would be right, okay? 10 years ago, I was on the phone with an FDA reviewer, and I was on the phone, so I'm not secondhand witness here, right? The reviewer said that a user could go to Costco or Sam's Club, buy the same brand of condom, and use the same brand of condom every day for the rest of their life. So they classified it as a permanent contacting device. Now, I've never seen that before, and I've never seen it since, but in this case, she wanted genotoxicity and subacute, subchronic toxicity on the condom. We were able to justify just doing an AIMS and a limited subacute, subchronic. We probably could have done some chemistry now, but back then that was not really what we went to. But this just kind of shows you that all of us can agree, but that doesn't mean that you're always 100% sure, right? What we always recommend, and maybe for a condom we would not have recommended this because it's pretty straightforward in my opinion, but if it's something complicated or unsure, we recommend getting a testing program together, submitting that to the FDA for feedback beforehand. Because you can get good feedback from them on an official pre-sub or Q-sub meeting and get an idea of how your design looks before you start initiating some of these long and expensive tests. Like I said, I probably wouldn't have done it with a condom because I thought everybody did the same thing, but sometimes we get surprised. Oxygen tube. What do you guys think? Okay, so this is huge in the industry right now. That's why I put it in here, because it's a little sneaky. Okay, so this person, it's hooked to a sleep apnea mask, and they don't make the mask, and they don't make the oxygen tank. They just make the tube. They didn't want to do any testing because they didn't have patient contact. We recommended and said, no, it does have patient contact, right? That was a couple years ago, so before that, we didn't realize exactly what kind of patient contact is considered. We got this response back from the FDA, which basically says that because of cumulative use of this over time, the oxygen acts as a liquid extract that can bring chemicals and particulates from the tubing set and deliver it to the lungs. So they consider it an external communicating device, permanent contacting. Okay? This hasn't changed. Although there are some who say they can get by with doing less testing, most of the gas path devices that we see are recommended doing this kind of testing strategy from the FDA. Okay? The reason I throw that in there too is to show that there are some changes that are happening with the FDA classification. And just because, once again, we might agree on something, it's great to check with the FDA beforehand before you go back. The last thing I'm going to talk about is changing of a colorant of an approved device. And the reason I put this one in here is because this is a very hot topic with the FDA. Anytime you have colorants or pigments in your device, the FDA is going to jump at it. I can guarantee it. So my recommendation is to stop coloring things. But that's not going to happen, okay? In fact, I just talked to someone yesterday that says, but I really want it purple. And I'm like, oh, why? So we can deal with it. There's some chemistry we have to do to evaluate the colorant, especially if you're on a liquid or gas path device. The FDA is worried about those colorants leaching off and going into the body. So if you have a colorant in your device that has substantial patient contact, either through indirect or direct means, then we can deal with that, but we have to have a conversation. Because there's some questions we have to get through. We have to look at the colorant, we have to look how it's used, the substrate involved, and get some really good idea of the possible biocompatibility impact. It'd be great if you don't color anything, but I understand if you have to. So tell your marketing people, if you're marketing in here, I'm sorry, but tell them you don't want it colored, and if they force you to color it, then come talk to the lab, okay? Okay, the last thing we're gonna cover is what happens if we fail a test. And the reason I throw this in here is because this is gonna happen if you do biocomp long enough. But I hate it when people freak out when they fail a test, especially cytotoxicity. I've seen people spend millions of dollars and tons of time developing a product, and they fail cytotox and they go, that's it, we're done, just throw it away. Please don't do that. A failed cyto doesn't necessarily mean the end of the world, okay? So there's some things we have to do though. First, we have to confirm the procedure, then we have to confirm the result, and either we have to eliminate or accept the toxicity of that test. So really quickly, I'm going to go through each one of these categories. The first one is basically just to make sure that the test is right. And if you work with a good lab, they'll work with you on this investigation. They'll look at maybe and I'm going to use cytotoxicity because that's the one that fails the most. They'll look at samples that were tested before and after to make sure there could be no cross-contamination. They'll look at the procedure to make sure that the cells looked good, everything was in check. But you also have to look on your end. Was it cleaned properly? Was it the right sample? I've had an R&D sample sent to me and the testing sample sent to the R&D lab. So it was all dirty and oily and things. So make sure everything is done correct, both on your end and on the lab end, okay? The next step is confirm the result. And this one is easy to do with cytotox because how quick and cheap it is. For something else, it becomes a little bit different. Because you don't want to just repeat until you pass, right? Because the FDA hates that. What we want to do though is we want to see if repeat tests have the same result. So we have to do more than just one. So we usually like to do 3 or 4 more tests to see if we see a true failure. The thing I recommend though is I want to see where the failure is coming from. So I want to break it down into components, but there's a secret to that too. If you send the lab 5 different components and don't tell them that it's part of a final device, they're going to treat each one as its own test, right? So they're going to do the surface area of that component and they're going to extract it in the same amount of fluid as that surface area. But that doesn't represent the final device because the final device is a total surface area. So what we want to do is take the 5 components and extract it in the same amount of fluid as the final device had. That way we can see how much toxicity each component is incorporating to the final device fluid. And then you'll know what component is the failing component. Because if I take a very small piece and I throw it in by itself, then it's all concentrated. But that surface area is very small in a final device, so it's probably not it can be contributing, but it's probably not your culprit. So by separating them out into individuals, but keeping the fluid consistent, the extraction fluid, you get to really see the total impact of biocompatibility. You can do some chemical profiling, see what comes off. This is great for metals, how much nickel is in there, how much silver, and you can see if that's a biological impact or if it's just a cytotoxicity issue. You can look at published literature. Have you ever had this issue before? Has someone else had this issue before out there in the device world? So let's say we found out what's wrong with it. We know it's going to fail. What do we do first? Well, the first thing you try to do is eliminate it, right? I mean, you might do some additional washes. You might have to change some procedures. Let's try to get rid of the toxicity because it's always easier to have a passing test than it is having a failing test. So let's try to justify. But let's say we can't replace. Well, the next thing is accept. And we do this quite often, especially with cytotox. Once again, latex. Latex fails all the time. But we can do a latex ELISA test to see how much latex there. We can do it compared against a predicate device like a latex glove or something else that has similar contact to be able to do dilutions on the cytotoxicity to find an endpoint. There's a lot of ways we can go about it to justify accepting the toxicity and then moving on and being able to accept the test. Okay, so I put some references in there for you, especially the 10993. So if you ever kind of want to know which one's -5, or you can look it up there. Any final questions? Okay.
The irrelevance of that test?
That's a good question. His question is, if cytotox fails, can you just argue that the test basically is stupid, right? I mean, that's the basic of it. It depends. So we can incorporate the sensitivity of the test in the risk assessment, but that's not what, you can't just say it's a bad test. And it's a very sensitive test, and a lot of times it's not relevant. But we have to be able to identify why it failed, make sure that that's not a reason for it to have clinical impact, and then accept it that way. So you just can't say it's no good. We can say it's no good, but this is why. So there's some investigation you have to do, and then you can say it's too sensitive a test. Did that answer your question? Good question. Great question. He says, in the extracts, what temperature do we use? In the ISO 10993-12, there's a range of temperatures you can use. It ranges from 37 to 121, and there's different time points for it. According to the FDA, this is the FDA's interpretation, if you have longer than 72-hour contact, they want 50 degrees or higher. And the reason why is because 37 is body temperature. This is Celsius because we're scientists, right? So we always talk about Celsius. So 37 degrees Celsius is body temperature. If it's longer than 72 hours, that's how long we extract. So the FDA is like, what happens over long term? So they want to elevate the extraction to look at worst case. Now, my recommendation is if your glass transition phase is not close to 50, so if it's 60 or above, then just extract at 50 degrees. Because my rule is always, I'd rather not talk to the FDA unless I have to, right? So if I extract at 50 degrees, I know I'm not going to get asked why you extracted at 50, or why you extracted at 50 degrees. But if I extract at 37, they're going to ask me, justify why that extraction temperature.
Is that correlated extracting at 50 is extended extraction? Because there's no correlation to say that. And if I have a chemical that's sensitive to temperature, and the FDA still wants me to extract at 50, right? Nobody's going to live at 50 degrees.
No, it's a good question. So two questions there kind of. One is the time kind of increase or decrease with the extraction temperature. It does a little bit. So 37 and 50 are both done at 72 hours. But then once you go up, the time gets less. So 70 degrees for 24 hours, 121 for 1 hour. So you do have a little bit of a correlation, but the 50 and 37 are extracted at the same time. The second question was about what if you have a temperature sensitive chemical? And if they extract at 37 based on that temperature sensitive chemical, but the FDA comes back and says, no, we still want it at 50. For me, we can fight that. Now, there are things that we want to look at. What kind of impact does the 50 degrees really have? We have to have a good scientific chemical justification, but the ISO is pretty clear when it says that you do not want to change the product for the extract chemically. So if the 50 degrees is impacting the leachability of the device or the chemical makeup of the device, then we can justify that.
My question is, we look at the
You get the microphone like me. Yeah.
We look at the ISO standards for guidance, right? But then the FDA requires additional testing on top of that. Do you have recommendations on how we can figure that out before the FDA gets back to us and says that the testing is insufficient?
Great question. And so what we give you is, so I'm going to go back real fast. So there are a couple answers to that question. One is if you see the diamonds here, these are they're tests the FDA may consider necessary. Honestly though, that's still up in the air because if your device is well categorized and well used materials, a lot of times the diamonds won't be asked for. But there are certain devices and certain contact uses that the FDA are concerned about. For example, we've seen a lot of personal lubricants asked to do a lot more testing than what the ISO says because the FDA is concerned about repeated use of a vaginal contact. So the easy answer is check the chart. The hard answer is you really need to have someone who you trust that you can just bounce something off to with them. So you just say, hey, this is my device, this is how it's contacted, this is the materials, are there any issues? And then if you have someone that sees this stuff routinely, because even though the FDA is trying to get more consistent, we still see some inconsistencies. And unless you're doing things regularly, you won't see those. So things change. And so just find someone that you trust, that you know that will give you good feedback and kind of bounce it off them.
Okay, thank you.
Uh-huh. Does that work? You have another question? Sorry.
Yeah. Do I need to do the device at the complete device and component level, both for the biocompatibility, or just at the complete device is okay?
It depends on your why you're doing the testing. So if you're doing it to get to the FDA and to do get a 510K or PMA, or then what you want to do is a final device with its final configuration, sterilization, all the processes. But if you really want to understand the device components, then we can separate them out. But the FDA is concerned just not about material, but it's about all the processing involved. So it's and the interaction between materials. So it's a final device.
If I change only small component of our existing device, do we have to do everything or do we only do the small part?
I get that question probably 3 times a day, okay? So here's my kind of thought process. We extract based off surface area, right? So the bigger something is, the more volume we add. And in the ISO 10993-12, in that table, there's a discussion about 10% differences in surface area, which means that if a lab does surface area and we do a surface area and there's a 10% difference in surface area, that we can still apply the results based off those differences. So I use that as kind of a consideration of impact of a change. If your change involves less than 10% of a surface area, then the impact to the patient is less, right? Because it's a less impact to the total of the device. Doesn't mean you just go, oh, there's no issue, but it means that we can evaluate using chemistry. We can look at how it leaches compared to the other one and prove that that small change didn't make an impact. And then we can use that to justify out of testing, or some of the testing, depending on the change. The bigger change you have, the more likelihood that the chemistry is going to be different between the two, and therefore you just have to repeat testing. So what I see is less than 10% usually doesn't have a big impact in leachables. 10 to 50% starts to see some impact. Anything over 50% is basically a new device, and we'll have to look at it from that point of view. But chemistry is a great way to look at a change and justify accepting it based off what comes off chemically. Good.
Does that work the same with recipes? So if you were to do a parent formula and decrease,
So I can say that again?
does that work the same with a recipe? So
With
formulations.
formulations?
Yeah.
Yeah, the same concept applies with chemistry.
Okay.
So it all depends on the formulation change and what impact it might have to the consistency of the material, what materials are in there, the leachability of the device. But chemistry can all be used to help justify it. So extractable leachables is a great way to justify out of changing because you can compare what leaches off of the original device and the new device to see the impact of the two. Any other questions? I think we have one more.
Oh, thank you. You know, when you have a new generation of devices and you use the predicator device, biocompatibility report, what should be considered and studied and evaluated to make sure that the FDA would approve that? Let's say through the years, let's say 5 years or so.
So there's a couple of trouble points I see with generations, because a lot of times what happens is there's small changes from generation to generation, and you start seeing, you did your biocomp on generation 1, and you made small changes that you kind of justified through the years, and now you're at generation 10, and you want to compare generation 10 to generation 9, but the biocomp was done on generation 1. So intermediate small changes can equal to big changes, right? But the rule is kind of the same concept, where what is the change? Where is it taking place? Is it taking place in the patient contacting portions? Is it taking place to a process that would impact the patient contacting portions? And then we can evaluate the surface area of that change and the impact of that change with chemistry to evaluate how much biocompatibility needs to be done. But I see a lot of people doing very limited biocompatibility from generation to generation, because most of the time it's like a mechanical change that might not impact materials, or it's a very small change that doesn't impact a lot of the biocompatibility based off the chemistry. Very good question. Okay, so if there are any other questions, we have a booth upstairs. Please come by, and I'd love to talk. I'll be here for a couple more minutes until they kick me out physically. So thank you for your time. You've been great.